this video, we will examine the comparisons between RNAi and CRISPR, as well as determine which one is best for finding mutations that have occurred in a specific gene. First, we must understand what RNAi and CRISPR are independently of each other. So, what is RNAi? RNAi stands for RNA interference. RNAi has two primary functions. The first is to regulate the expression of certain genes in the body, and the second is to limit the intrusion of pathogenic antigens in the body. It does this by shutting off gene expression using double-stranded RNAs to limit or inhibit translation in the cell. This is a quick overview of how that is done. First, dsRNA is cleaved by the enzyme dicer to form miRNA or siRNA that is about 21 NTS long. miRNAs and siRNAs bind with proteins to form an RNA-induced silencing complex that base pairs with mRNA and either inhibits translation, miRNA, or degrades mRNA, siRNA. Some potential positive uses of RNAi include in medicine and in agriculture. Now you might be asking, what is CRISPR? CRISPR stands for Clustered Irregularly Interspaced Palindromic Repeats. It is a prokaryotic mechanism that's function is to cleave invading DNA. Modern use has adapted CRISPR to target other genomes to allow for specific genome editing. CRISPR-based genome editing requires two components, a guide RNA and a CRISPR-associated endonuclease protein, in this case Cas. The guide RNA directs the Cas nuclease to the specific DNA sequence, which then cuts the DNA at that site. Cas9 creates a double-strand break in the DNA, which the cell repairs using one of its natural mechanisms. What are some of the main differences between RNAi and CRISPR? A primary point of distinction is that RNAi reduces gene expression at the mRNA level, which is equivalent to, let's say, a knockdown, while CRISPR completely and permanently silences the gene at the DNA level, which is equivalent to more of a knockout. Also, CRISPR has less error. Even though both systems could find a mutated cell in a certain gene, I would choose RNAi over CRISPR for this specific task. Although CRISPR is generally more accurate than RNAi, it doesn't make up for the fact that RNAi is semi-permanent in nature, whereas CRISPR permanently alters a genome. The mutation in the gene hasn't specifically been said to be good or bad, and I wouldn't want to risk permanently damaging something beneficial.